Hi, everyone. Welcome to our Tuesday seminars on the cerebellum. June Liu is our guest today. June is one of the leading places, June's lab is one of the leading places in the world that studies the effect of trauma and stress on the brain, and she focuses on the cerebellum. So as you know, trauma causes anxiety, depression, drug addiction. She studies the neurobiology of stress on the cerebellum. June received her MD from Beijing University and then came to America to do a PhD in biophysics at the University of Rochester. Then she did a postdoc at Yale with Len Kasmerek, who trained her in electrophysiology, and she worked on the expression of potassium channels in the cerebellum. Then June went to University of College London, where she studied with Stuart Candy, who trained her on the study of AMPA receptors in the cerebellum. And the idea was that stress and other trauma alter the neural activity so that they change both the neurons and the glia, and the glia then has an effect on synaptic transmission and regulation of the neurons. So June's going to tell us about the role of the cerebellum and control of plasticity for emotional memory.
Can I start?
Yes, please.
Okay. All right. Thank you, Reza. I'm sorry for the introduction and for the invitation, and as well for organizing it, really, because I've been learning a great deal from this seminar series. So today I'm going to talk about the cerebellar circuit plasticity that controls the emotional memory formation. So cerebellar cognitive affective syndrome, which is also called Shadmehr syndrome, was first defined by Shadmehr and Sherman in their early paper in 1998, in which they described that a patient with lesions involving posterior lobe of the cerebellum and the vermis exhibited behavior change. And these include impairment in the executive functions, such as planning, and in the spatial cognition, that including the visual, spatial organization, and memory. They also have the affect deficit in the affect regulation, such as inappropriate behavior and the personality change, as well as linguistic processing deficits. At the end of the paper, they think that a constellation of deficits is suggestive to disruption of cerebellar modulation of neural circuits that link prefrontal, posterior parietal, and the superior temporal and limbic cortices in the cerebellum. And some of these predictions has already turned out to be true. So one of the animal model that has been used to study emotional learning and memory is Pavlovian fear conditioning. And here the animal will be exposed to a tone, and then which is co-terminated by a brief foot shock. And such that the animals start to learn that these two events are associated with each other. And one day or days later, the animal can be placed in a different context, different environment. And then when they hear the tone, the same tone while they were during the learning phase, they will display a freezing response, which is a defensive response, and suggest the animal remember that the tone will predict a subsequent food shock. The work done by Sacchetti in 2002 have shown that after the training fear conditioning learning paradigm, they injected tetrodotoxin into the cerebellum to inactivate the activity of cerebellar neuron. In this graph, they show it's in the cerebellar cortex, And then that injection can occur 15 minutes after the fear conditioning, or 24 hours, 3 days, a week, or 2 weeks later. And then 2 days after each tetrodotox injection, they look at the freezing response to the tone. So that's a memory retention test. And the animal received saline injection is showing the white bar. They show a prominent freezing, which about 80% are freezing to the tone. However, those animals received a TTX injection, they show very little freezing. And this impairment of memory recall actually lasts within 1 week of the fear conditioning. If you inject TTX, it will cause a different degree of impairment of memory retention. However, 2 weeks later, there's no effect. So from this, they conclude that cerebellum is important for the consolidation of associated fear memory. So the question we want to ask is, how does the cerebellum control the fear memory formation? As we mentioned before, cerebellum form extensive connection with the cortex, with limbic systems. Therefore, cerebellum can probably act as a relay center for the sensory information. And the neuroplasticity that is important for memory formation actually occur outside of the cerebellum. This has to be said. There are evidence supporting this model. For example, the paper published in 2023 in Cell Reports by Huang from Lee's lab, they show that DCN neuron to PBN parabronchial nucleus connections actually is important for memory formation. And this connection also undergo learning dependent plasticity. And then there's a paper from Frontera in 2019. They show the connection with PAG is also important for memory formation. But here we are trying to look at whether the cerebellum itself is the site of memory formation. So to do that, the fear conditioning needs to induce a lasting change in neural activity within the cerebellar circuit. And the second, this type of cerebellar plasticity should be required for the consolidation of fear memory. So if these two requirements are met, then the cerebellar fear conditioning induced change probably can encode the memory formation. So that's our goal of the project. So cerebellum, we all know about our focus actually is on the inhibitory interneurons. So Purkinje cells receive input from parallel fibers and molecular layer interneurons, as well as climbing fibers. And it projects to deep cerebellar nuclei and the PBM. So conditioned stimuli, which is tone, could activate the multi-fiber that activate granule cells and therefore parallel fibers, whereas unconditioned stimulus, such as a food shock, will activate the climbing fibers. The co-activation of these fibers will lead to an increase in the excitatory synaptic transmission at the parallel fiber to Purkinje cell synapse. So Sacchetti and his colleague in their 2004 paper showed there's this potentiation. They think it's important for memory consolidation. And more recent study by from Lee, I think, it's and the colleague, they have shown that if you selectively knock out the STAT3 from Purkinje cells, that will cause an increase in the synaptic transmission at the parallel fiber to Purkinje cell synapse. And this animal show an enhanced memory retention. So it's consistent with the concept that potentiation at this synapse is important for memory formation. And the Selfo and the colleague in 2008 showed that fear conditioning also enhances GABA release onto Purkinje cells. So the question we ask is, how does this fear conditioning alter synaptic transmission, especially in these molecular layer interneurons? We look at the endocannabinoids. So endocannabinoids, they are lipid molecules. They are important neuromodulators. These neuromodulators is known to play an important role in the extinction of fear memory. Its role in the memory consolidation was still a little bit controversial based on some of the pharmacology studies. So stellate cells, these inhibitory interneurons and the parallel fibers both have endocannabinoid receptors. They actually express a quite high level of endocannabinoid receptors. Within the cerebellar circuitry, it has been shown that both Purkinje cells and the molecular layer interneurons can produce the endocannabinoid because they have the DAG lipase, that enzyme that synthesizes 2-AG. And the 2-AG is one of the major endocannabinoid in the cerebellum. And once it binds to the receptor, once it's released, it binds to the receptor, it can suppress GABA release and glutamate release. And endocannabinoid then can be removed by an enzyme called MAGL lipase, so that terminates its action. So we first take a look and see whether fear conditioning can alter the level of endocannabinoid in the cerebellum. And this work is done by Chris Dubois and Jessica Patel and several other people, and it was published 2020. So here we use several protocols. So the animal is naive group, and another control group we have is we use fear conditioning protocol, but it's unpaired. That means it gives 4 tones, and then 30 minutes later give 4 foot shock. 8 tones and then 8 foot shocks, but they are separated for about 40 minutes, such that the animal do not learn any association between these 2 stimuli. About 1 day later, they will take the cerebellum out and then remove the lobule 1 to 6, and then process it, extract the lipid, and then subject to the mass spec, and then quantify the 2-AG level. So the 2-AG level in the naive animal and unpaired animal, they are about the same. But after fear conditioning, the level was reduced. If we normalize this 2-AG level to the naive control, we will see actually unpaired actually has about a 20 to 30% decrease into overall 2-AG levels. And then we look at whether the endocannabinoid signaling is also altered after fear conditioning. So the early study have shown that the lobule 5 and 6, they are important in fear conditioning, and the lobule 9 and 10 are not. So right now, the first experiment we look at is we look at in lobule 5 and 6, whether endocannabinoid level, the signaling is reduced. So we voltage clamp stellate cells in the vermal lobule 5 and 6, and then measure the miniature iPSC. And each of this downward deflection is an inhibitory current. And you can see, and then we apply a CB1 endocannabinoid receptor antagonist, which is NES. You can see this when we apply NES, the number of iPSCs increase. So therefore, the frequency of miniature iPSC frequency actually increased, probably nearly doubled. And the same thing happened in the unpaired animals. However, after fear conditioning, first we notice is the miniature frequency in the unpaired animals are higher than the naive control and the unpaired control, which is shown here. They are higher than these two. And secondly, when Chris applied NES, actually the iPSC frequency no longer increased. So from the results like this, we conclude that fear conditioning can reduce endocannabinoid signaling. It occurs in the lobule 5 and 6. This is just to reiterate the signaling, how endocannabinoid was metabolized. It's usually released from postsynaptic cells upon the membrane depolarization or activation of G protein. In the cerebellum, activation of metabotropic receptors can evoke endocannabinoid release where the activating GQ protein. And then once it's released, it can bind the receptor and then suppress neurotransmitter release. And then the endocannabinoids can be taken up by astrocytes, in this case, Bergman glial cells, or the presynaptic terminal, like parallel fibers, where they will be undergo degradation because they have the enzyme called MAGL lipase. So we just showed that the 2-AG level was decreased after fear conditioning. So there are two possibilities. One possibility is the endocannabinoid production, it was reduced after fear conditioning. Second possibility is the degradation of endocannabinoids was enhanced. So we start to look at these two possibilities. To separate the production from the degradation, we use an approach called depolarization-induced suppression of excitation. We call it DSE. You can also measure depolarization. You induce a suppression of inhibition. That will be called DSI. So using DSE as an example, here Chris patch stellate cells and then stimulate the parallel fibers. Now, each time you stimulate parallel fiber, you will evoke an EPSC recorded here. So after a stable recording, he will depolarize stellate cells. That will cause release of endocannabinoid, which will suppress, hopefully, the neurotransmitter release. So after the depolarization, he will continue doing the evoke the EPSC, evoke the EPSC. Immediately after the depolarization, the EPSC amplitudes are much smaller compared with the prior to the depolarization. And then if about 10-15 minutes later, 10-15 seconds later, the evoked EPSC amplitude start to increase compared with the immediately after depolarization. About 30 seconds later, it almost back to the control level. So he will plot, normalize each of these amplitude to the average control level. Okay, And then after depolarization, you will see the amplitude becomes smaller. It's about half, less than half of the initial amplitude. And with time, the amplitudes become greater. About 30 seconds, they are very close to the initial level, the baseline. To show that this depression is due to activation of endocannabinoid, he will include endocannabinoid receptor antagonist NES in the solution and then repeat the same experiment. Here it shows before the depolarization and this after depolarization. Now it no longer shows the depression which exhibits in the absence of the antagonist. So therefore, we can use the magnitude of depression often reflect the production of endocannabinoid and the receptor sensitivity, and the time course of the returning to the baseline or recovery. It's often a measure of endocannabinoid degradation. So now we go back to the experiment. Here we only show the unpaired group and the fear condition group. As you can see here, the gray open dots are the measurement of DSE from this unpaired animal, which shows just like naive animal, about a 50% depression. At about 30 seconds, it returned back to the completely recovered. However, if you look at the fear condition, which is this black circle, you will see it actually have the same level of depression. The maximum depression is about the same. But what difference here is it returns back, recovers faster. Even within 10 seconds, it almost back to the original level. So if you fit this with exponential fit, you will see this one has a much faster recovery time in the pair condition, in the fear condition than the unpaired control. To show that this difference is really due to the degradation of endocannabinoid, he included JZL in the solution. So JZL is a MAG lipase inhibitor. By putting the JZL in, you knock out all the MAG lipase activity. In this case, you will see, so the naive animal and the unpaired, the paired animal, actually they have the similar time course. They both are very slow now. So we think that fear conditioning here really shorten the DSE, and therefore it accelerates the degradation of endocannabinoid. To show that it really affects, this can be affecting MAG lipase activity or expression. So we look at the expression level now using the immunohistochemistry, and then we stain for MAG lipase immunoreactivity. Again, both unpaired and paired training on day one. Next day, it was sacrifice the animal and then do the MAG lipase immunostaining. If we look at the lobule 5 and 6, which is supposed to be involved in the fear conditioning, we saw that there's a immunostaining in this is molecular layer and the granule layer. And MAG lipase are known to express in granule cells. Therefore, you see a little bit of punctate staining here. In the molecular layer, it will be parallel fibers have some immunostaining, and the Bergman glial cells process. Therefore, it's a little bit of diffused staining. Now, after fear conditioning, we see a prominent increase in the molecular layer. MAG lipase immunoreactivity compared with the control. So when we quantify it, we do see a large increase in the MAG lipase immunoreactivity after control. The black bars are for the lobule 5 and 6. If we compare, this is in the molecular layer. When you quantify granule cell layer, there's no difference between unpaired and the field condition control, animal. So then when you look at it in the same slides, you quantify the MAG lipase expression in the lobule 9 and the 10. The first thing you notice is here in the molecular layer, the immunostaining is much higher compared with lobule 5 and 6. And that's turned out to be the case. If you look at the lobule 5 and 6, and this is lobule 9, 10 in the molecular layer, we saw a quite large increase in the immunoreactivity. And the second thing is when you compare unpaired control with the field conditioning, there is no difference. So it looks like field conditioning selectively increase MAG lipase expression in the lobule 5 and 6, and not in the lobule 9 and 10. So from this data, we think we can conclude that actually field conditioning does increase the endocannabinoid degradation by increasing the MAG lipase expression, therefore accelerate the degradation of 2-AG. So the next thing we wanted to look at is, does this change in the endocannabinoid signaling is responsible for memory consolidation, cerebellar-dependent memory consolidation? Because the endocannabinoid release can be evoked by activation of GQ in Purkinje cells. So we tested whether we can use a DREAD, GQ DREAD, to evoke endocannabinoid release in Purkinje cells as a way to manipulate cerebellar endocannabinoid release only in the cerebellum. So this is the L7 Cree GQ DREAD animal. So we breed L7 Cree with a GQ animal. And then you can see here, the that's YFP is only expressed in Purkinje cells. And then when Chris did a recording, look at stellate cells and the Purkinje cells. So these are extracellular recording. These are the spontaneous action potential firing in stellate cells and Purkinje cells. Then he applied 1 micromolar of CNO, which is the agonist of DREAD receptors. Then he found that it didn't really affect the stellate cell action potential firing, but it changed Purkinje cells action potential firing from regular activity to this bursting activity. So the next thing is to test whether CNO application can cause an increase in endocannabinoid level. That's what we really want to do, right? We want to selectively increase endocannabinoid level in the cerebellum. See how does that affect memory retention. Again, we used a similar approach. We measured the miniature iPSCs from stellate cells, and then we apply CNO to the cerebellar slices, and we waited for 30 minutes, and then we measure the iPSC, miniature iPSC again. If you look at the downward deflection, the number of downward deflection after CNO application is reduced, which is shown here. However, if we include a CB1 receptor or endocannabinoid receptor antagonist in the solution and repeat the same experiment, now we will see that actually application of CNO no longer reduce the frequency of miniature iPSCs. So this experiment shows that activation of GQ dread in Purkinje cells from this L7 GQ animal can cause release of endocannabinoid. So in the behavior experiment, what he did was you subject the animal to fear conditioning. 30 minutes later, inject CNO. We do this because we want to CNO has effect only during the consolidation period, not during learning phase. And then next day, the animal will be put in a different environment, a new context, and then play the tone. And this is the freezing response we observed. When the animal, so we have both GQ positive animal and GQ negative as a control. When these animals receive saline injections, both exhibit freezing, about 60% of freezing, indicating they can remember that the tone is associated with the shock. However, the animal receiving CNO show a different pattern. The GQ positive animal actually show a much reduced freezing to tone compared with the GQ negative animal. So activation of GQ can impair the memory formation. And then on day 3, he tested contextual memory retention. That is, the animal will be put in the same box where they did fear conditioning. The only thing is there's no tone and no foot shock here. And then the animal, if they can recognize the environment, they will freeze, and both phenotype actually freeze about the same amount. So given the fact that activation using CNO to activate Purkinje cells can induce a burst activation, so one possibility is endocannabinoid release can cause this impairment of memory retention. Another possibility will be this burst activity of Purkinje cells can cause downstream, can cause neuroplasticity downstream of Purkinje cells and possibly even outside of the cerebellum. So to test this as a possible possibility, Jessica actually inject the animal with a CB1 endocannabinoid receptor neutral antagonist. It's called AM4113. Immediately after fear conditioning, 30 minutes before the administration of CNO. And now on day 2, when she tested the acute memory retention, now the GQ positive animal no longer show this a decreased impairment of memory retention. So this experiment really suggested the endocannabinoid release from Purkinje cells is responsible for impairment of memory consolidation. So the results we have shown so far, I think, showing that learning can increase the MAG lipase expression, which reduce endocannabinoid level, tonic endocannabinoid level. And then this can cause, and because the decreased endocannabinoid level will reduce the endocannabinoid suppression of GABA release, which is like a disinhibition, and then that increased GABA release, and then probably that causes memory involving memory consolidation. So now our question is the second part. So does cerebellar interneurons control fear memory consolidation? So this work is done by Katie Cazzoli, Georgios Kogias, and Jessica Patel. So Jessica actually started with trying to inhibit molecular layer interneuron, see whether that causes the memory consolidation problems. So here she will use NOS-3 mice, and the NOS is and nitric oxide. Since it's in the adult, it's only expressed in the molecular layer interneuron within the cerebellum. So she will, excuse me. She will inject the GI dread virus into the lobule 5 and 6 within the vermis. And then, so later, after 2 weeks, she will prepare the slice, and then she can show that the M-cherry actually is only expressed in this part. It's a molecular layer interneuron. That's the granule cell layer. That's the Purkinje cell layer. And then this is a zoom in. These are all stellate cells. When she recorded action potential from one of these M-cherry expressing neurons, she found that they do show spontaneous activity as the regular stellate cells do. And the application of CNO will cause a membrane hyperpolarization. You can see the membrane, the base decrease, and also silence the stellate cells. So stellate cells no longer fire action potentials. So with this knowledge, she conducted a behavior experiment in which they injected a GI dread, a virus encoding GI dread, or M-cherry only as a control. And then 2 weeks later, the animal will be subject to fear conditioning. And 30 minutes later, will receive either CNO or saline. And then the memory retention will be tested the next day. So we look at the M-cherry receiving CNO, GI dread receiving CNO, and the GI dread receiving saline. They all show freezing during learning. And on the second day, during the memory retention test, both control M-cherry CNO mice and the GI dread, and with the saline injection, they show freezing about 50%. However, the GI dread that receiving CNO show almost no freezing. We were actually at the beginning very surprised, and then we repeated it. It's exactly the same every time. In the 3 animals, we missed the lobule 5 and 6. And these 3 animals, which now shown here, also show about 50% of a freezing in the memory retention. So it really is a lobule-specific effect. So this molecular layer and interneuron activity drives memory consolidation. So if that's the case, well, wouldn't the fear conditioning increase molecular layer interneuron activity? Therefore, that can drive the memory retention. To test this, we first look at whether how this molecular layer interneurons, the passive membrane property has changed. So in this case, we did a voltage clamp, whole cell voltage clamp experiment, where we actually give hyperpolarizing current injections in the naive animal. So we measure the changes in membrane potential. In the black trace is the naive animal. And then you will see the membrane become hyperpolarized. At some point it stopped and it's become depolarized. And this is called a voltage sag. And it's a characteristic of an expression of IH currents or HCN currents, which is activated by hyperpolarization and is sensitive to cyclo-AMP, cyclo-GMP, cyclonucleotide. It's an inward current. Usually that's caused depolarization. So if this sag is really caused by IH current, then the inhibition of IH current should abolish this. Indeed, when we apply ZD, which is an IH current inhibitor, the hyperpolarization becomes bigger and we no longer see the sag. After fear conditioning, the changes in membrane potential in response to hyperpolarization is more like control with ZD. That is, cause a greater hyperpolarization, and the ZD has no effect. And importantly, this sag actually disappear. So from a similar experiment with a smaller hyperpolarizing current injection, we calculate the input resistance, which is from the current injection and the voltage response. We found that the fear conditioning increased the input resistance, almost double the input resistance compared with the naive. And the second, in the naive animal, application inhibition of IH also increase the input resistance. So an increased input resistance will suggest now the neurons are more responsive to synaptic input. So the next thing is well, before just mentioned, there's also change in the membrane potentials. The resting membrane potentials was measured, and just by breaking into the membrane, the cell, in the control, we see it's about -60. However, after fear conditioning, the resting membrane potential become more hyperpolarized, closer to -70. And inhibition of IH currents also cause an hyperpolarizing shift in the resting membrane potential in naive, but not in after fear conditioning. So these experiments suggest fear conditioning actually indeed change the passive membrane property of the stellate cells. To test now what happened to the hyperpolarized ramp current injection and the subsequent action potential firing, which is a rebound activity. We inject the current, ramp currents of 50, 100, and -150 picoam. In the naive animal, you see when you inject the negative currents in, it causes hyperpolarization. And the end hyperpolarization, it fires action potential. Now, after fear conditioning, the hyperpolarization becomes greater than the naive animal. And what's also interesting here, at the end of the current ramp injection, the action potential firing is higher than before. So this is like a rebound potentiation. Now, if we apply IH current blocker to the naive animal, you will see the same thing happens. It increases the hyperpolarization and it enhances the rebound activity compared with naive. So again, these results together with the passive membrane property shows that the fear conditioning actually increase the molecular layer interneuron response to hyperpolarizing input. And now Katie, look at the response, molecular layer interneuron response to excitatory stimulation. So first, she did a current clamp recording, injecting a 45 picoamp currents into stellate cells. In the naive animal, it evokes action potential firing. And in the fear conditioned animal, there are two things happen. First, you probably notice there was a delay in the naive animal before the first action potential fire upon depolarizing current injection. But after fear conditioning, this delay become a lot of shorter. So the first spike latency is much reduced compared with the naive animal. And the second thing is the action potential firing frequency is a lot of higher after fear conditioning than the naive animal, which is shown here. So this is the action potential firing in the naive animal, and after fear conditioning, there's an overall upward shift in the frequency. And when she applies ZD, which is IH current blocker, they found that ZD can also shorten the first spike latency in the naive animal, shown here. And it also causes an increase in the action potential frequency, just like in the fear conditioning, which is shown here. So fear conditioning really increase molecular layer interneuron response to depolarization. Now we are looking at the molecular layer interneuron response to parallel fiber stimulation, which will be more physiological. We look at, we did a extracellular recording on the stellate cells, and then stimulate the parallel fiber with a different stimulation strengths. Here is an example of at 6 volts. And so it gives 4 stimuli, each of them evoke an action potential. In the naive animal, we will quantify the action potential firing within 100 milliseconds after the stimulation. So naive animal evoke some action potentials, but in the fear conditioning animal, it evokes more action potentials after this burst stimulation. This is a group data. It shows in the naive animal, we show slow increase in the action potential firing, whereas after fear conditioning, which the action potential frequency is much, much higher compared with the naive animal. So the stellate cells response to the excitatory stimulation is much enhanced after fear conditioning. So now we start going back into IH currents, because the implication of previous experiment is IH currents is important. So we now directly measure the IH current in the voltage clamp recording. This is a typical voltage clamp recording. So this part is called the instantaneous currents, which is a leak current. And then at this point, you see a slowly activated outward current. This is IH current. IH currents activate slowly. That's why when in the earlier experiment, we show a rapid hyperpolarization. And then slowly, when the IH currents start to activate it, the membrane potential become more depolarized, because these currents are inward currents, and it causes membrane depolarization. This is after fear conditioning. And then she used ZD to inhibit the IH currents. And this is the difference between the two currents they measured. So she called this ZD sensitive currents. That means it's IH currents. So here it shows fear conditioning reduce the IH currents. You can see in both the current trace and the group data. And this change occurs about 1 day afterwards, and Giorgio later on tested even 7 days later, the IH currents remain reduced. So this is a long-lasting decrease in the IH currents. And then if you look at the conductance voltage relationship, and this is a relationship that tells you how at which potential the currents will be activated. In the naive animal, the half activation is about -70 millivolts, but after fear conditioning, it shifted leftward, become more hyperpolarized. That means you need to go to -80 millivolts in order to activate half of the channel. So make it more difficult to activate it. Therefore, the currents measured at -120 at all these potentials are smaller compared with naive animal. And this only occurred in lobule 5 and 6 when they measured the current, IH current at in the lobule 9, they did not see any difference after fear conditioning. So this effect, it's lobule specific, only affect the lobules that is involved in the fear conditioning. Okay, so the work, the previous work done in the Saltis lab in their 2016 paper, they show that in the hippocampal neurons, the IH currents can be regulated by endocannabinoid. So we start to ask question, since we know endocannabinoid level decrease after fear conditioning, we ask the question whether the decrease in endocannabinoid can lead to a decrease in IH currents. So
May I? Yeah, June. I want to give some time for questions. So if maybe we can, just to let you know, we're running out of time.
Okay, I will speed up. Okay, so to do that, we actually look at IH currents. That's Giorgio's work. And in naive animal, when you apply CB1 receptor antagonist, it blocks the currents. In the fear conditioned animal, the opposite happened. You apply CB1 receptor agonist, it enhanced the IH current. And so that suggests that endocannabinoid regulate the IH currents. A decrease in endocannabinoid after fear conditioning actually leads to a decrease in the IH currents. He then include a G beta gamma subunit inhibitor, GALAN, in the patch electrode. Repeat this experiment. This inhibitor completely block the increase in the IH currents upon the application of WIN. So this is the results. So the data here has shown that endocannabinoid activate the receptors, and then through activating G beta gamma subunits, it will activate the Jun kinase and then nitric oxide synthase that will cause the increase of cyclo-GMP and activate the IH currents. And very briefly, we went back to the early study so to see whether activating GQ dread in Purkinje cells, it's also affect the IH currents. In the GQ negative animals, we show a different decrease in IH currents, and then the CB1 receptor antagonist didn't do anything. Whereas in the GQ positive animals, instead of having a suppressed IH currents, these animals show impaired memory retention, also have a larger IH currents. And these currents can be inhibited by CB1 receptor antagonists, just like in the naive animals. So this work suggests that endocannabinoid released from the cerebellum during the memory consolidation can abolish IH plasticity. So in conclusion, here we have shown that fear conditioning can actively increase MAG lipase expression and reduce 2AG level. And then that's important for memory consolidation. The activity of inhibitory interneurons actually is a driving force for the formation of memory consolidation. And this decrease in the 2AG can reduce IH currents and then increase the response, steady cell response to inhibitory and excitatory input. And we think that contribute to the memory consolidation. In conclusion, we think a cerebellum is the site of memory formation. And thank you. And these are the people who did all the work. And thank you for your attention. I will take questions.
Thank you so much, June. So if you have a question, please just go ahead and raise your hand. I will start with my question. You began by showing that in that PNAS paper, the consolidation lasted a few months, and then at 1 year, performance of the animals was the same as control. So
this is about the 2 months what they did. So these, I'm sorry, these are days. No, hours. I'm sorry.
Hours. Okay.
This was hours.
So if I understand it correctly, then at 380 hours, there is no effect anymore. So where is the memory at that point?
It is probably suggests the memory itself is not stored in the cerebellum, but there is a period the memory is processed and then transferred to the other brain region. So it's involved consolidation, but not the storage itself.
Right. Yeah. Thank you so much. Are there any other questions for June? Maybe I can go with another question. So we know that this part of the cerebellum, lobule 5 and lobule 6, is of course is involved in many kinds of behaviors, facial movements, arm movements, whisking. How does fear memory, if it produces these changes there, how are those changes not affecting other behaviors?
Well, we didn't really test other behaviors. I don't think we can really state it. We did test some like open field, and then we didn't see any change. But in terms of yes, we haven't really tested other motor function other than open field in our study. So it's possible there are other changes that we just haven't really tested.
Well, I think the changes you have found in MLI response are very exciting, and it connects with what Jason Christie was showing a couple of weeks ago.
Yes, yeah, they did look at the motor, right? I couldn't really attend that one, his talk. Yeah.
It was a VOR task that they studied.
Yes.
What's the future for this work, June?
Well, we are sort of expanding it to other aspect of fear memory. And we also look more into the coding aspect, see particularly which type of neurons are important in encoding the memory formation in the lobule 5 and 6. So it's more of looking at identify the neurons who are activated during learning or extinction, and then see whether this type of neurons actually, how the memory excitability or neuronal activity change. And
June, what role do climbing fibers play in learning of this task?
so we haven't really looked at just in vitro activated parallel fiber. So the assumption, I think, is a climbing fiber is without a climbing fiber activation, this type of plasticity wouldn't happen. And the earlier study, they also show that climbing fibers actually can innovate, not synaptically innovate the stellate cells, but they, through this spillover, can activate the stellate cells. So that's I think I believe that during learning, it's the co-activation of parallel fiber to stellate cells, and together with the climbing fiber spillover activity, that together can induce the plasticity in the molecular layer interneuron.
Interesting.
Yeah, and because it has been shown that Purkinje cells themselves, the intrinsic excitability didn't change after fear conditioning.
Well, thank you so much. Thank you, everyone. It's a pleasure to learn from you, June. Thank you so much.
Thank you. Bye.